Hi guys, this is Mr. Wayt, and I'm sorry I can't be there with you today. I wanted to go over just a few things with you really fast. For your journal today, if you open that up in AP Classroom, find the quiz BRNR 151, Structure Function of DNA. Take a look at that. There's only one multiple choice question, and when you answer it, go and record your answer. Remember, I'm not worried if it's right or wrong. But talk about it with a friend next to you. Go ahead and see what they think, see what your table thinks, and then maybe as a class you can kind of come up with an answer together. I'm going to go ahead and recommend you pause the video now, think about it, get this question, and I'll talk about it here in just a little bit. All right, I hope you've had a chance to look at that question, record your answer. Let's go ahead and review 151, and then I'll tell you our plan for today. So really quickly, let's start out with this one, and we will get to the question in the next slide here in just a little bit. But this says linear sequence nucleic acid is defined by 3' hydroxyl, 5' phosphate group of the sugar in the nucleotide. So we're looking at a monomer of nucleic acids. This is the most simplest unit, the monomer, and a nucleotide always has phosphate, sugar, base, phosphate, sugar, base. And we've talked before about on this 5-carbon sugar right here, we have one, we'll name each of those carbons 1', 2', 3', 4', and 5'. Now, it's the 2' carbon that either has the H or the OH, the deoxy or not deoxy, but it's this 3' down here and 5' up here that are opposite of each other. The 5' is always connected to this phosphate group right here. So when we look at DNA, looking at the nucleotide, we can see there's a direction to it. If 3 is on the bottom, 5 is going to be up top. So let's take a peek at the next slide that might help us with the answer to your question that we looked at earlier. DNA is structured anti-parallel. Anti-parallel means one goes up, other one goes down. If I could do something like this, kind of hard, man, I can't get a place to see it. Anti-parallel. One goes 5 to 3, the other goes 3 to 5. They're held together by hydrogen bonds. Now, this is the key thing right here. A always pairs with T, C always pairs with G, but when A and T, adenine and thymine pair, there's always only 2 hydrogen bonds. So if I were to anneal or to break these, or sometimes they say denature the two, separate these two strands, A to T with only 2 hydrogen bonds would be simpler and require less energy than looking at C to G. C to G has 1, 2, 3 hydrogen bonds, and because of that, it would require a little bit more energy to break these bonds than it would the A to T bonds. Guys, one little jingle we have to remember that, and it's not great, but I got this from another teacher, and this goes like this. AT is a 2-letter word. C to G is 3. I know C to G is not a word, but if you can remember, AT is a 2-letter word, C to G is 3. That might help you memorize the number of hydrogen bonds in between those. Hey guys, this also says during DNA synthesis, nucleotides can only be covalently added to the 3' end of a growing nucleotide. So something else you might want to add to your notes. 5 to 3 is the way to be. So you'll notice down here, it always is going to be added from the 5' towards the 3'. Now don't forget this blue one is anti-parallel. So if this one is 3, this one is 5. And this dark one right here is the new strand. Now don't worry, if you don't get it this time, we're going to see this again in future units. But when we add new DNA, 5 to 3 is the way to be. Even down here, don't forget it's anti-parallel. So this time we have the bright blue is 3, 5 over here. So we'd make the anti-parallel. 5 to 3 is the way to be. We'll talk about why these are segmented later. All right, 5 to 3 is the way to be. Excellent. If you change this linear sequence of nucleotides, you're going to change the information coded to build proteins. And remember, proteins make pretty much everything. So the example that we have right here is this DNA sequence is exactly what it should be. But this one down here, we're going to exchange You see this one has a CTG, this one is CAG, and because of that, that's going to change both strands. What that does is it codes for a different amino acid. Instead of glutamic acid, we get a valine. And now you can see that results in sickle cell, which we'll talk more about later on. But that one small change in a DNA nucleotide has profound effects on the organism. So something we'll talk about later as well. Guys, I hope you have a good day. Before I do that, let's talk about your plan for today. Don't forget, all this is posted on Google Classroom. I've updated the bill table of contents as well as the lesson plan. So guys, after you did this today, you have the rest of this period to work on biologically important molecules. If you're not finished with that, I imagine people still need time. And if you do get finished, you have time to work on the properties of water activity stations. That's going to be true for tomorrow as well with another assignment. Guys, let me just take you to the properties of water activity stations and remind you what that was the last time. And I know it's hard to think when we did this over a week ago, but in station 1, here were the directions, and here's where you made your observations. If you flip the page over, what I ask you to do this time is to write an explanation, not an observation, and you must include these words. So station number 1, part A, this is the one where we were building a molecule with the kits, a molecule of water and a molecule of methane. So if I come down here and I'm supposed to explain what I observed, and I have to use these two words, at least those two words, I might say something like, because water molecules are polar, that enables them to form hydrogen bonds with other water molecules. I remember in the lab, I said I would tell you how many water molecules could be bound to one water molecule. And the answer to that is 4. You can look up pictures of that if you'd like. But don't forget, we have a positive end and a negative end, and opposites attract. So if you'd like to see that, I'd be glad to show you when I get back. I do plan to be back on Thursday. If you guys have any questions at all, please make sure you email me detailed question, and I'll do my best to get back to you. I will not have internet except for the evenings. So when I sit down, I'll try to get to emails. Guys, I hope it goes well, and I'll look forward to seeing you when I get back.